is Alex Kentsis. I'm a pediatric oncologist and cancer biologist at the Memorial Sloan Kettering Cancer Center. My laboratory science is inextricably linked to my practice in pediatric oncology. For some time now, I and many others in the field have been trying to understand how young people can develop cancer when they otherwise have not been exposed to mutations in the environment or have not inherited defects in genome integrity that would cause cancer mutations in the first place. A major source of evolutionary innovation are jumping genes that can mediate the creation of new genes and new functions in all living things. And on occasion, they've become co-opted for normal functions. A few years ago, in studying the composition of childhood tumor cells, we found at the time a new uncharacterized human gene called PGBD5. We identified PGBD5 to be highly expressed in a very unusual type of a human cancer called rhabdoid tumors. These are rare but terribly lethal cancers that affect young infants and children. This is the first time that an endogenous human DNA transposase has been found to mediate DNA rearrangements in tumor cells, thereby explaining a long-standing puzzle in the field about how mutations can occur in otherwise healthy young children without exposures to mutations or inheritance of DNA instability causing lesions. My research leverages the latest instrumentation in mass spectrometry proteomics and functional genomics to define at high resolution the composition of cancer cells and what makes them different than normal cells. In a sense, what makes cancer cells in their own right. And with those tools to identify the fundamental defects and liabilities that cancer cells, in particular those with activated DNA transposases, have in order to target them with new emerging targeted therapies. This work would have been impossible in as close as 10 years ago because the ability to define the molecular composition of cancer cells and to understand the mechanisms by which their genomes are rearranged can only be done with recently developed methods in functional genomics and functional proteomics. With the support of the Pershing Sohn Prize, we're now able to rapidly pursue questions relating to the fundamental origins of childhood cancers, and in particular, to try to develop knowledge that will allow us to develop new improved therapies for children with refractory tumors. My hope is that over the next few years, we'll be able to understand in detail how this process is regulated in normal cells, how it becomes dysregulated in cancer cells, and with this knowledge to identify new, precise therapies that restore normal control to achieve cures for the vast majority of children and young adults with cancer.